final speaker of this session is Dr. Svante Pääbo. Pääbo was the first scientist to successfully sequence the genome of Neanderthals. He also discovered the previously unknown hominin group Denisovans. Recently, Pääbo and colleagues found evidence of a first-generation child with mixed Neanderthal and Denisovan origins, providing evidence of interbreeding in humans of different subspecies. Pääbo is the director of the Department of Genetics at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. Today, he will share his insights on the future of ancient DNA. Please welcome Svante Pääbo.
Okay, so thank you very much. It's very exciting to be here. I was then asked to talk about the future of ancient DNA, which sounds like an oxymoron to me. So I sort of allowed myself to add one word at least in the title. And what I actually then wanted to do is to base this on work that has gone on in our lab over the past 15, 20 years on extinct forms of humans. But what I will be discussing and doing sort of some cautious extrapolations into the future will apply to any other organism, such as mammoths, that you might be interested in, from which you can retrieve DNA. But we have then this sort of obsession with Neanderthals. To the left is a reconstructed skeleton of a Neanderthal, to the right of a present-day human. And as you know, Neanderthals, they evolve in Western Eurasia and Europe to Central Asia around 400,000 years ago and live there until approximately 40,000 years ago when they become extinct, in conjunction with that modern humans appear on the scene. And one may then ask, why should we be so interested in Neanderthals? I think there are two reasons for that for me. One is that they are the closest relative of all present-day humans, evolutionary relatives. So if we want to define ourselves genetically and biologically as a group, it's really them we should compare ourselves to in the first instance. And another question may be how present-day people are related to Neanderthals. But to then do that, you need to retrieve DNA from the remains of Neanderthals, and that's then something that we worked on in the meantime almost 30 years in our lab. And there are issues with that. First of all, that there is very little DNA surviving in bones of ancient remains, often 100,000, a millionfold less than in fresh tissue. It's degraded to very short pieces. It's chemically modified. And it exists in a large excess of bacterial and microbial DNA that colonized these remains over tens of thousands of years. And as a consequence of that, contemporary traces of DNA from present-day people, humans say, if you study Neanderthals, that will not at all affect your experiments if you study fresh DNA, may totally overwhelm your experiments when you study these tiny amounts of DNA in old bones. So in the meantime, ways to overcome that has been worked out. Part of it involves sort of being really paranoid about cleanliness, working in special laboratories, wearing funny clothes, etc. And also methods with which you extract the DNA efficiently and cleanly and transform it into forms where you can then do DNA sequencing and other analysis. So in the meantime then, we have several Neanderthal genomes from different places in Europe we can study. We also have some very good Neanderthal genomes from 3 different sites in the meantime. So these are then genomes where we have gone over the last few years to have some fragments covering a part of the chromosome here, to having, in average, every site covered 30 or 50 times over. But I should stress that that is for the approximately 2/3 of the genome where we can map these short fragments. There are still repetitive parts of the genome where we don't know the exact arrangement of DNA sequences and probably never will for these extinct species. So we can then relate these Neanderthals to each other. The oldest one is in the order of 120,000 years, the youngest one's 40 to 50,000 years ago. And one discovery that then came out of comparing these genomes to genomes of present-day people was that for many of us today, we carry fragments of Neanderthal DNA in our chromosomes. So one chromosome here indicated in red, and you carry between 1% and 2% of your genome from Neanderthals if your genetic roots are in Europe or Asia or anywhere outside Sub-Saharan Africa, actually. And just as an example of that, for example, a particular variant here that's associated with high levels of LDL cholesterol. It sits then on a big piece of DNA of 130,000 base pairs that is almost identical to the Neanderthal genome, and it's really the size of this that then proves that this comes over from Neanderthals quite recently, so it hasn't had time to get smaller due to recombination in each generation, so it couldn't come from back here from the common ancestor. So since everyone outside Africa carry these traces of Neanderthal DNA, also in parts of the world where Neanderthals have never been, the model that explains this is then that when modern humans come out of Africa, they probably met Neanderthals quite early on, perhaps in the Middle East. And if these humans mixed with Neanderthals, they could carry with them this Neanderthal component, so to say, out to parts of the world where Neanderthals also had not existed. And these sort of remnants of Neanderthal DNA in the genomes of many of us today have consequences. As you will see here, different people, each line is a different individual, tend to carry different fragments of the Neanderthal genome, and they turn out to often be associated with things that can affect us today. Just two examples. There is, for example, one variant here of a gene that is quite frequent in Asia. Up to 20-25% of Asians carry it. It clearly comes from Neanderthals. This is the Neanderthal variant here. These individuals in Asia are closely related to that for this gene, which encodes a lipid transporter that's associated with risk to get type 2 diabetes, the type of diabetes you get at old age. Just 2 weeks ago, there was another paper that appeared that showed that a progesterone receptor variant that exists particularly in Europe and Western Eurasia comes from Neanderthals, and that's associated with an increased risk of preterm births. And there are many, many such cases now for diseases and sometimes protection against these diseases where Neanderthal variants influence the risk. So say hypercoagulation, there are many skin diseases, even things such as depression and so on. But what has made all this progress then possible is improved methods to retrieve tiny amounts of degraded old DNA from small amounts of material. So today we often work with 40-50 milligrams of bone powder. And a sort of striking example of that is this little bone. It's a fragment of the last phalanx of a finger that was found by Russian archaeologists in 2008 at this site in southern Siberia, at the Denisova cave. And we were able to sequence quite a good quality genome from this finger, and was very surprised then to find that it was not a modern human, was not a Neanderthal, but something else that went far back to common ancestor shared with Neanderthals, that's about 4 times deeper back in time than any split between two present-day populations among humans. So this was clear it was some new group of extinct hominins that we then called Denisovans after this site in Russia. And still today, this group of individuals is only known from this little fragment and 3 teeth that has been found in this cave. So it's a group of hominins that we really only know from their genome sequence and not from any fossils that one has found. Yet we think that they've been quite widespread in the past in Asia, because all over Asia, people carry small amounts of Denisovan DNA in their genome, and people in the Pacific, say Papua New Guinea, people have up to 5% of the genome from these Denisovans. So we then think that when there were Neanderthals in Western Europe and modern humans appeared, there were these Denisovans probably in rather large parts of Asia. So this is then part of a trend where you can now retrieve DNA, not just from big, nice bones, but from very tiny bones, and also from other types of material. So, for example, from coprolites, sort of old poop that you find at archaeological excavations, dental calculus, for example, and what's particularly interesting at the moment for us, from ice cores and from sediments in the permafrost particularly. So we've focused very much on that lately, looking at archaeological sediments from archaeological excavations, using tiny amounts, and trying to retrieve DNA from them. And that is then often possible. For example, in this site where these Denisovans were found, one can then show that deep down there are Denisovan remains, DNA in the sediments, and Neanderthal DNA, Denisovans again, Neanderthals, and so on, reconstructing the history of who has lived in this cave. And if you want to do this on a large scale, you of course need to process hundreds, if not thousands, of soil samples. And that now becoming possible in terms of that DNA extraction and the production of these libraries, immortalized forms of this DNA that you can study, and the capture of the parts of the genome you're interested in have all been automated. You can do it with robots, such as this lab robot here, and go into a cave and actually study hundreds and hundreds of sediment samples through the stratigraphy. And for example, reconstruct then very well what animals have lived at different times in this cave. And you can also do it then for hominin DNA and find, for example, Neanderthals in these samples here and Denisovans in these samples further down. And so far, this has only been done on mitochondrial DNA, which is a tiny part of the genome that exists in many copies per cell. So it's particularly easy to retrieve. But the big picture, of course, if you want to reconstruct the history of individuals, is the nuclear genome. So very recently, we then focused on that, trying to make DNA probes to fish out interesting parts of the nuclear genome from these sediments. And the challenging part with that is that there is much more animal DNA there. So you need to then do probes to fish out this that excludes all relevant animals, allows you to focus only on hominins here. And that turns out to be possible if you sort of target parts of the genome where, say, humans are unique and other hominins are unique in having some substitution where they differ from everyone else. You will preferentially fish out this, and you will also recognize that you have right DNA molecules. So you can then, for example, distinguish very well modern humans from Neanderthals, from bone DNA, go into archaeological sites, take sediments, and see that in one of these sites there were clearly modern humans that lived. In the 4 other sites, there were Neanderthals. So I think as a first extrapolation into the future then for the next decade or two, I think this will become common practice, presumably in archaeology. You may just drill cores at archaeological sites or maybe even across the landscape and isolate DNA from different depths down in the stratigraphy. So you will have truly molecular archaeology, if you like, not limited to looking for bones or artifacts anymore. So you can have these cores, and you can, at different points in this core, sort of then fish out, at the moment, a bit over 1 million sites in the genome. And if you then take only one DNA fragment representing every such site you're interested in, you have sort of a statistical population sample about what that population looked at genetically. So you will then be able to sort of use that to sort of reconstruct population history across time. And the beginning of that sort of exists, but by studying bones, as you may know, if you now go across time and look in Europe, where most of this work has been done, on certain time periods from which you have DNA genomes retrieved from bones, you can begin to go across time and follow population changes across time. And that's then something that will presumably become much more common with the sediment DNA in the future, I believe. The other area that I then want to stress here is that when we now have the genomes, for example, of Neanderthals, our closest relatives, we can then focus on genetic changes that happened here recently in human history, after we separated from Neanderthals. And we are particularly interested in that, of course, because it might be sort of the genetic background of functions unique to modern humans that hide there. And that may be interesting because with modern humans, of course, comes technological change that happens very fast, comes things as figurative art that really depicts things, and a tremendous increase in population size, and populations start colonizing all habitable part of the planet, influencing in the end the whole biosphere, as we heard in the last talk. So what one can now do then, when one has these good genomes from our closest relatives, is to focus on the things that changed there and became present in all people today, no matter where we live, and make a catalog of those things. And to me, the fascinating thing is that that's not a tremendously long list of things. It's in the order of 30,000 changes in our genome. And I think a very interesting direction in the next decade or two is to focus on those changes from a functional perspective, to try to find out, if you like, what made human history possible in terms of a biological background. And that is work that's just sort of in the very infancy. Just to give you a feeling for that, how one might go about that, let's focus on the most simple changes of those 30,000. There are 96 changes that change amino acids in proteins. They affect only 87 genes. And if we just look on one of them that we have started studying, this ADSL, that encodes an enzyme involved in purine biosynthesis, so the synthesis of two of the nucleotides in DNA and in many other functions in the body. This change look interesting because it's close to the active site of the enzyme. So what one can then do, and what we have done, is to engineer this change in human stem cells back to the ancestral state. And you can then compare the metabolism of these wild-type normal cells that we all carry in our bodies with these ancestralized cells, ancestralized for this function then. And you can then look in the metabolism downstream of the two reactions catalyzed by this enzyme, and you will find that the human cells, the concentration in the cells of all the metabolites downstream are lower than in ancestralized cells. So this change then obviously happened, at least to a large extent, after we separated from Neanderthals. You can look at other genes here. A striking thing is actually that there are 3 genes here that encode proteins that are all involved in the spindle. So that's the molecular machinery at cell division that pulls the chromosomes apart to the daughter cells. It seemed very surprising that such a sort of conserved basic mechanism might have changed very recently in human history. But we have some indications of perhaps what might be going on. And that comes from other work that goes on in our department, where we study brain development, comparing humans to apes. So that's then work where you use stem cells from humans and apes. You can make those stem cells make these brain organoids. So these are 1-2 millimeter big little bubbles which mimic sort of early stages in brain development. And you can compare that then between, say, chimpanzees and humans. And overall, it's very, very similar, but there are some subtle changes that you can actually find. So if you look in the epithelium here, there are stem cells at the top of the epithelium, which is down on this picture, that divide and make eventually neurons in the cerebral cortex. So if you now look at time-lapse images here of chromosomes during cell divisions in humans and chimpanzees, you will see the chromosomes lining up here and then being pulled apart during cell division, here for the chimpanzee, and you will notice that that happens a bit later for the humans. So this phase when the chromosomes are lined up before being pulled apart is longer in humans than in the apes, and it's specific, it seems, to these stem cells during neuronal development. So obviously that makes these 3 genes particularly interesting. So what you then want to do is to go and engineer these changes into human stem cells back to the ancestral state and make these brain organoids. Also engineer this into the mouse, the modern human changes into the mouse, and study brain development. To then find out if this happened back here or if it happened earlier back there. So this is then an area where we're sort of working on doing this, we and others, sort of investigating the metabolism, as we indicated, the cell division then, aspects of how cells establish, neurons establish connections to each other, and other areas you could think about. And obviously, what will happen in the future, I think, is that one will get more extinct forms of humans, not just Neanderthals and Denisovans. One will have other forms here. So it will become possible then to study not only time changes, not only see what happened here, since we separate from Neanderthals, but going back and looking for things that happened there, and know what happened even further back there. So you will time when these changes happen in the past. Something that will also happen is, and is already beginning to happen, is that we have many Neanderthal genomes. So we'll also be able to see what was fixed among all Neanderthals, the things that happened here and define Neanderthals as a group. And we can similarly, of course, test those in stem cells and in model organisms. So to sort of then end up, I think that what one can then do is sort of recreate phenotypes of extinct organisms, perhaps metabolic pathways, perhaps organelles in model system that we already heard hinted at in the last talk. And what we also heard hinted at is that there are people who are more visionary, who go around and suggest that we should even bring species back, even Neanderthals back, and that's of course something that we will not be doing. But to end then, I think the sort of things that I think will happen over the next 10 years or so in this field is that we will have a truly molecular archaeology, where one will then be liberated from being limited to just finding bones and artifacts. One will be able to sort of isolate DNA at archaeological sites just from sediments and perhaps also across the landscape. One will be able to do truly population genetics over time because one will have many, many genomes from many, many places from many different time periods, hopefully systematically over the an area. So you will be able to not just use models to see what happened in populations in the past, but you will actually be able to observe, if you like, in real time, genetic variants spreading in a population and becoming fixed in species. And finally, what will be really interesting is to reconstruct past traits, and that will then be done in model systems of different sorts, cellular model system and organismal model systems. And there are, of course, many, many people that have been involved in this, more than I can mention. I will just show the faces of the current group leaders in the department that all have been involved, are involved in some aspects of what I've been talking about. There are also groups that we collaborate with, of course, particularly Wieland Huttner and his group on brain development in the mouse, and Philip Kajtovich on the metabolic analysis. And with that, I then thank you for your attention.